Hello, everybody. We're here for lecture 14 for Biochem 440, covering intermediary metabolism. Hopefully be a shorter one. There's a couple slides I want to go over, really, but the rest of them I don't really think are that important. So go ahead and get started. So, the first slide I want to take a look at um, is just a nice way of showing that it's very complicated. Nothing really that important to it. It's just know that they're all very interconnected. These are some definitions you should know, just generally speaking. So anabolism is generation of molecules, and catabolism is breakdown of molecules. So like the breakdown of glucose is a catabolic reaction, and the buildup of glucose to give to the rest of the body from the liver, for example, would be an anabolic process. So anabolic is buildup, and catabolism is a breakdown. Um, there's anabolic pathways that build up things like proteins, polysaccharides, lipids, all this stuff from these amino acids and sugars, all this stuff. And there's catabolism, which takes these carbohydrates and then turns them into these different things like ammonia, like waste products, and then ATP. So basically, anabolism uses up ATP in these energy molecules, and catabolism creates these ATP molecules. Yeah, so the idea here is that catabolic pathways, right? Catabolic pathways are breaking down things to turn to energy, so they all funnel into these energy molecules, right? While anabolic ones kind of use those energy molecules to create other things, right? So they're used to make other things. So like ATP, for example, is used for other cellular processes in your body, or to use to make proteins, or used to make sugars, or used to make lipids, stuff like that. Why do you want to learn about metabolism? Very important for the human body. If you're taking this course, biochem major, I assume many of you, maybe med school is the intention. So obviously very important for that. These hormones will show up extensively, like insulin in type 1, type 2 diabetes, all that jazz. Like liver failure, all sorts of um, interesting organs that rely on metabolism to basically do their job. Um, So yeah, this is an interesting one. It's basically showing you as the duration of activity goes on, what is the most like probably like uh what's the highest kind of activity used to create energy, right? So you've got pre-existing ATP, phosphocreatine, glucose, and then glucose again, right? So glycolysis and glycolysis for and then respiration. This slide's not that important. I've never seen them test on this, so I wouldn't worry too much about it. Again, I don't think this is that important. I guess what you should know about this is like as time passes, you use up ATP, you create this creatine phosphate, goes back down, and then glycolysis, and then yeah, not something just too important. Yeah. So why study metabolism again? So a lot of human diseases, right, are based on uh metabolic issues. So like enzymes being broken down. I think phenylketonuria is an example of this. You've got this kind of retinal degeneration here, which is through IMP dehydrogenase, which is an enzyme that actually takes the precursors of purines and turns them into a xanthine monophosphate. So you got inosine monophosphate and then xanthine monophosphate. So we basically take this pre-matured purine and then we turn it into a another form here. But basically, if you mess up this enzyme, that doesn't work anymore, right? So you don't really make these nucleotides in those cells. Phenylketonuria, right? I think this is phenylalanine hydroxylate. I can't remember off the top of my head the actual name of the enzyme, but basically, if this enzyme doesn't work right, you can never produce tyrosine, and tyrosine is used for a lot of things, if I'm not mistaken. There's other kinds of diseases as well, like all these are glycogen storage related, and if you don't store glycogen, right, very difficult to exercise is really difficult. You need glycogen stores for when you're not eating food, right? Because you're not always eating food, right? You have to store some of the energy, but if you have trouble with storing them, um, yeah, you can't really do that. And you'll learn about all of these enzymes for glycolysis on Monday. Cancer metabolism. So, cancer is usually mutations in different enzymes cause a tumor growth. Um, I wouldn't be too worried about this. I guess the one thing to know is that they will sometimes ask you about these different metabolic pathways. So they can make up pathways right on exams and test you on it. So like, what kind of reaction is this going from isocitrate to alpha ketoglutarate? Well, it looks like some sort of oxidation reaction, right? You got this oxidizing agent NADP plus, and you're also releasing CO2, so a decarboxylation reaction, right? And then going from alpha ketoglutarate to that, there's a mutant version of it that takes it to 2-hydroxyglutarate, which is an oncometabolite. Which basically an oncometabolite, from what I understand, is something that basically promotes cancer. Yeah, so I don't know. When I took this class, didn't really have to know a lot of these. I guess the one that you did need to know is pulse chase. So this guy right here. And then, of course, metabolic blocks and inhibitors is just like relatively straightforward. So we're going to cover a couple of these. So starting off with RNA-seq. So RNA-seq is very simple. Basically, what you're going to do is you're going to take your cells, right? You're going to extract the mRNA from these cells. And the premise behind this is that different cells will express different RNAs, right? Because of epigenetic modifications, the location of the cell in the body. All your cells have the same DNA, right? But obviously, your liver cells are different than your skin cells, right? So even though they have the same DNA, the RNA products that are actually being made from those cells are actually very different. Right, so the basic idea is that you're going to take a cell, right? You're going to extract the mRNA from it. You're going to fragment this mRNA up, or you're going to complementarily form the DNA to it. So you basically do PCR on it, right? And then you basically read these sequences, and then you match them to the human genome using using software of some kind, right? And what you get is these exonic reads, where these mRNA sections that are actually being produced, right, end up showing up on and you can basically map them here. So basically this shows you, right, that there's a lot of RNA with this section of nucleotides. There's a ton of RNA with this section of nucleotide, this section, like all of these are being expressed in this cell, right? But notice how these sections here are not being expressed at all, which means that this cell does not require that this section of nucleotides or this section of nucleotides, right? But the idea here is that we're basically sequencing the different RNAs in the cell, and certain cells do not require certain RNAs. So like this RNA here, in this section here, might be necessary in a different spot, like a different cell type, right? And just not in this one. Basically, the main gist here, we take mRNA, we turn it into cDNA by doing PCR on it. We cut it up, right? And then we basically read it using Sanger sequencing or next-gen sequencing or something like that, right? And then when we sequence it, we map that sequence data to the human genome or to some sort of genetic library, right? And we see which reads show up the most. For proteomics, it's basically just we take the protein. This can be done in many ways, right? You can do SDS page, you can do size exclusion on it, which you guys have learned about. You can do immunoprecipitation, which is basically just taking antibodies and binding those antibodies to the protein, right? And then you digest them, you separate them out, and you use mass spec from OChem. If you remember, basically you take this protein, you fire beams into it, and it breaks up the protein. And then using that mass spec data, you can actually construct the protein's structure from it. I don't think this is that important for your exam, but it is here, so just be aware of that. Gas chromatography, GCMS, so gas chromatography mass spectrometry, right? Basically, you have this metabolite that you're extracting. You're going to put this into your machine here, and what's going to happen is it's going to try and boil it or evaporate it, right? and then you'll basically get these different reads, right? So it's basically actually a 3D axis. So like you'll get, so remember at this time point right here, right, you'll get a lot of product being released, right? And then you have the mass spec going in the Z direction here, the negative Z direction, right? So the idea here is that you do the same thing as you did up here, right? Where you basically take the protein, you digest it, right? The only difference is this time is you're actually like boiling it basically, or you're gas chromatography, right? You're basically boiling it, right? And it's going through this detector, right? And this detector is picking up the different proteins and different molecules in the what's it called, in the solution as they pass through, right? And we have a big intensity here for a lot of boiling, so a lot of molecules have been released, right? And we have a lot of molecules here, and then here's the mass spec for it. So you can basically identify what this protein is based off of how much of it is there and what the mass spec looks like, right? Okay, so NMR spectroscopy, you guys did this in OChem. It's a bit different here. It's a little more 3D. Basically what you can see here is that as you go, I think it's easier to look at this graph a little bit, right? So we have ATP, we have inorganic phosphate, and then we have phosphocreatine. So you can see that over time, like ATP levels are actually relatively constant. So these black dots here, right? These guys, like this section here, it's actually very constant. It's like pretty flat. What is changing though is the phosphocreatine levels. So like when you're doing exercise or something, right, you can see that the phosphocreatine levels actually drop, and then what actually ends up being released is the inorganic phosphates from phosphocreatine. So what this basically says is that during these like exercise states or muscle contraction states, is that we're actually using phosphocreatine and not ATP a lot of the time. And basically, how are they doing this? Well, you remember NMR from OChem. This is very a 3D version of that. Again, I don't think it's important to know this for the exam. I think this is just extra info. I think there's better things they could test you on, but basically what they're doing here is you've got the ppm, right? And you've got the different, they're basically looking at these phosphate groups, right? So they're actually looking at phosphate. This is PNMR, right? By the way, P31 NMR. So they're basically looking at the chemical shifts of the different phosphate groups, and they're trying to see where they're at. And they notice that with time on the z-axis here, right, that there's a big dip right here that comes back up, which means that the phosphate is being released, right, and then put back. That's the main gist of that. And here's that pathway if you want to see it a little bit more. So I think, yeah, so pulse chase is the one that's kind of, I think, a little bit more important. So you can do other things as well. You can do C14 detection. They actually do tend to, like, when you guys start getting into the pathways, they will like label a carbon and ask you to track that carbon through the cycle. That is something they tend to do. It didn't show up on my exam, but that does not mean it won't show up on your exam, right? So so yeah, so you can basically say but you can use carbon, right, to figure out how much metabolite you have, right, in your system. So for example, if you don't know, you obviously don't know how much labeled metabolite there is, right, unless you have a labeled amount, right? So there should be 100% total metabolite, right? So if you know how much labeled metabolite there is, you should be able to figure out how much unlabeled there is, right? And basically detect it using mass differences using mass spec, right? So this C13 adds an extra like atomic mass number on top of the molecule, and that is detectable using different techniques that I don't think, again, are that important. So move on. Yeah, so this is where I would actually like start paying attention. So pulse chase is something they can test you on. So basically, right, you see this graph, right, and let's imagine you don't see this down here, right? You should be able to tell, right, what the pathway is. If you look at this, compound A starts at 1,000, compound B starts at 0, right? And what's happening is that as compound A decreases, compound B is increasing, right? And there's a midpoint, right, where they both meet, right? But what that means is that A is being used up to make B, right? And you should be able to predict this pathway given this data, right? Or given this pathway, you should be able to predict what the data looks like, what the graph looks like, right? So the idea here is that since A is decreasing and B is increasing, and they're increasing at the same rate for the same like duration of time, they're probably just a direct pathway that goes into each other. The other way to kind of sanity check yourself is to take 2 points on this graph and just check the math, right? So your total should equal 1,000 or whatever the number is up here, right? If we look at time 25 or something, or like 20 right here, let's say, right, we can see that the radiation here is 250 for compound B, and it's around 750 for compound A, and 750 plus 250 is, lo and behold, 1,000, right? So they should add up to equal the total max at the very top of this. So, I believe, yeah, so if you look at this, right, how would you interpret these results? This is obviously very different from this. So compound A is decreasing, right? And then compound B is also increasing at the same rate. So you might be thinking, oh yeah, well, it's just going to be A to B and that's it. Well, not quite. Because if it was just A to B, this should extend and just keep going up like this. But the problem is it's actually going down eventually, which suggests that there is another thing that is being made after B, and we just have not found it. So basically, this is most likely going to be A to B to C, or something along those lines, right? And you can double check that using your math, right? So if we look here, we've got, again, like around 250 here and around 750 here. So that's 1,000. So there's clearly none of this third molecule being made at that point. Let's check here. Let's look at 50, right? This is probably around, I don't know. Okay, let's see. Is there a better number? You want to try and pick numbers that are easy to find. If you try and do numbers that are a little bit harder, it can be a little bit challenging. Let's see here. I guess we can try and do this. This is a bit of a weird number. I honestly would not recommend maybe doing this, but this is probably around 350 if I had to guess, right? This is around 500, right? So seems like there's 150 missing, right? And 150 would be probably around like this area here, if I had to guess, right? And you can basically do this for every single point, right? And if you go down here, maybe you can look at this. This is like 150, and this is like 2 something, and that's basically going to be 300 something. Basically, maybe it's around 700 or 600 something. So maybe around here, right? So you can see how I'm starting to build this out, right? And so basically, if you do this right continuously, there's 250 here, 350 here, 550, there's 450 remaining, so like maybe there's a little bit right here or something, right? You can see that I'm starting to build out this curve and it looks something like this, right? You could basically go from there, and lo and behold, there you go, right? Basically, you just do this math method, right? And you can build out these curves yourself, right? We guessed this pathway already, right? Because we saw this trend in B that was going down, right? Even though like A is technically also going down. Because if it was a single transition here, only this and not this, you would just expect it to go whoop and just go up, right? But because it's not doing that, there's some third metabolite in there. So now we can also do the same thing for this, right? I won't go into depth about this, but basically this one's a bit harder to notice, but what you should be able to do is if you add up the numbers, right, you should notice something's weird. So like here, for example, at this point here, we're at 300 and 300. We're missing 400, which is really whack, right? So here, we're going to put a point there. Maybe if you do like, let's see here, like 200, like here, this is 200 and right here, and then like 600 there. So like another like 400 here or something, right? And then maybe if you check here, it's like 150 maybe, or like 100 and something maybe, and then 600, so like 700 here, so maybe like 300 here, right? So you can basically, again, you can keep doing this, right? And you can build out this curve. And you can say, oh, and it does this, and then eventually drops down or something like that, right? And you can eventually build this out, and you get this molecule here as X, right? Basically, you just got to do that over and over again until you get the answer, basically. So, steady state versus equilibrium metabolism. So, the idea here is that with equilibrium means that there's no net flow. So the rate in is equal to the rate out, right? So these two are equal and opposite, right? That's what equilibrium is. There's no net flow, right? That's the main thing there, right? The steady state is a little bit different, right? In the idea that the input rate and the output rate are equal, right? But the rate of the forward reaction does not equal the rate of the reverse reaction. So these two are not equal, right? But it seems like the reaction is in equilibrium because the rate of this and this is the same, so it only looks like you can see these two rates here, if that makes sense. There's still stuff being outputted and still stuff being inputted, but because their rates are equal and opposite, so the rate of inputs going into A and the rate of outputs from B to this output here are the same, they basically cancel out, and what you're left with is just this intermediate area here where we're going from A to B. And in that case, the A going to B is faster than B going to A, right? But that's not the whole story of the equilibrium, because there's actually stuff going into A and there's stuff going out from B, right? Hopefully that makes a little bit of sense. Is there anything else I want to say about this? Forward reaction. I feel like there is something else I want to say about it, but I'll make a comment about it, I guess, in the description if I have something else to say. Yeah. I guess the idea here, right, is just if I have a large pipe, right, and some amount of water, right, it takes a bit longer because there's a large pipe and there's a lot of area for the water to go through, right? If I have a smaller pipe, right, the water only has certain places it can go, right? So it's much faster in a smaller area than a larger area. I believe this is actually one of the last slides. Yeah. Okay. So inhibitor analysis. So this is also something that you should be able to analyze and understand. So basically, right, we've got this we've got these metabolite concentrations, right? And you've got these enzymes, and you have an inhibitor, right? So, but you don't know what the order is of these. You don't know if A goes to B, goes to C, goes to D, or if maybe it goes from D to B to C to A or whatever, right? You have no idea what the order is. So, what you do is you inhibit an enzyme, right? And you see which which one decreases the most, right? So you say you know that an inhibitor is known to inhibit a certain enzyme, right? And you add this inhibitor, right, into these cells, right? And what happens is that you produce like 0D, but you still have a bunch of buildup of A, B, and C. So what that tells you is that this inhibitor that you added, this enzyme, right, that you just added this inhibitor for, is probably turning something into D, right? And that is the very last step of the reaction, right? If I added an inhibitor, right, and then this C right here was like dropped way down here along with the D, these were both like down here, right? That would tell you that it's probably inhibiting the second to last step. So it's probably inhibiting from B to C, right? basically you can tell, right? So whichever ones are lower, right, means that the enzyme that's making that product is actually inhibited, right? So if I inhibited B, or it's like if I got rid of if it was B, C, and D where all three of them were low, right, and A was very, very high, like A was up here or something, right? That would mean that the inhibitor you added is most likely affecting the first step of this process because A is massive and the rest of them are small, right? So basically, you can tell which enzyme is being inhibited and where that enzyme is in the metabolic pathway based off of the amount of metabolite concentration available. That wraps this up, though. So yeah, so I would say like generally you should know what information we get from different techniques. Um, I guess like what are the advantages and disadvantages of them, and then um for these, so for pulse chase and inhibitors, right? These are the main one you should know for your exams, in my opinion. These are very easily testable. Flux is just like a concept from Bio 220, right? Yes, this idea that flux equals gradient divided by resistance, right? You have a very high gradient, right? You'll have a lot of flux. If you have very high resistance, you'll have a little flux, right? So just something to keep in mind from 220. But yeah, and then obviously identifying these metabolic pathways based off inhibition or pulse chase experiments, right? So please, please, please practice using these because these are what's most likely going to show up on your exam. But other than that, I hope this video was helpful. That is the end of this one, and thank you for watching.